Today in this video we are going to talk about three important topics. One is we'll talk about the growth media, and another will talk about special culture requirements. We'll talk about various bacterial cultures, and we'll discuss about the various type of bacteria: aerobes, anaerobes, facultative anaerobes, and various intracellular bacteria. We'll talk brief about these types of bacteria. So first of all, let us talk about the growth media. So there are two properties of the growth media. One is the growth media could be the selective media, and another is differential media. The differential media is also known as the indicator media. So first of all, let us discuss about the selective media. So the selective media favors the growth of particular organisms, and it prevents the growth of other organisms. So one of the examples of the selective media is Thyromatin Agar media, which contains antibiotic that allows the selective growth of the Neisseria by inhibiting the growth of other sensitive organisms. So one of the examples of the selective media is Thyromatin media. That means Thyromatin Agar. So for the explanation of the selective media, I have here shown you the two different culture medium. On the left you can see the chocolate medium, and in the right you can see the Thyromatin medium. Here the specimen is the rectal sample, which is used for testing for Neisseria gonorrhoeae, and you can see in the chocolate medium is that there is overgrowth of the various bacteria. But the Thyromatin media, I have already told you that the Thyromatin media or the Thyromatin agar contains the antibiotic that allows the selective growth of Neisseria by inhibit the growth of other sensitive organism. So in the Thyromatin media there is the growth of Neisseria only. So there is the specific colonies growth that occurs in the Thyromatin media. You can see in the picture also. In the chocolate medium there is overgrowth of the various bacterial colonies, but in the Thyromatin media there is growth of the Neisseria bacteria. And another is the indicator media, or we also call it as differential media, because we call it as a indicator media because it changes color to donate to indicate that there is the presence of the bacterial colonies. So this type of media yield a color change in response to the metabolism of the certain organisms. So for example, let us say the McConkey agar, which contains a pH indicator. A lactose fermenter like E. coli will convert the lactose to acid metabolites, and which change the color to pink in the McConkey agar. So these are the examples of the differential media and the indicator media. So what I mean to say that McConkey agar contains a pH indicator. So the lactose fermenter bacteria like E. coli will convert the lactose to acid metabolites. So those acid metabolites will change the pH indicator towards the pink. You can see in this picture also, I have shown the two group of colonies in the McConkey agar. One is lactose fermenting colonies, another is the non-lactose fermenting colonies. So you can see that lactose fermenting colonies of bacteria will turn the color of the McConkey agar into pink, whereas the non-lactose fermenting colonies will be the colorless. Now let us discuss about the special culture requirements. What are the special culture requirements for the various bacteria? So now first let us discuss about the chocolate agar. The chocolate agar, the detailed preparation of the chocolate agar is not covered in this topic. If you want to know the detailed preparation of chocolate agar, then you have to read some of the microbiology books. But in the brief, I want to tell you that the chocolate agar is formulated by heating the blood agar in the certain temperature, and the main content of the chocolate agar is the factor 5, which is NAD plus, and the factor X, which is the hematin. And the chocolate agar is specifically used for the Haemophilus influenzae. For the culture of the Haemophilus influenzae, the chocolate agar is used. And now let us discuss about the Thyromatin agar. The Thyromatin agar selectively favors the Neisseria by inhibiting the growth of gram-positive organisms. So it contains various antibiotics like vancomycin, trimethoprim, colistin, and nystatin. You have to know that the gram-positive organism is inhibited by the vancomycin, and gram-negative organism with trimethoprim and colistin, and the nystatin inhibits the fungi. So it is the special culture media for Neisseria gonorrhea and Neisseria meningitis. So the Neisseria species are specifically used for the Thyromatin Agar. So the Thyromatin Agar, as I have said is previously, it is the selective media. So basically the Thyromatin media is chocolate agar plus the antibiotics that I have told you. The antibiotics that are vancomycin, trimethoprim, colistin, and nystatin. You have to take in mind that the vancomycin will inhibit the gram-positive bacteria, whereas trimethoprim and colistin will inhibit the gram-negative bacteria, whereas the nystatin will inhibit the fungi. Now let us discuss about the boarded Django agar and Regan-Lowe medium. The boarded Django agar and the Regan-Lowe medium are specifically used for the Bordetella pertussis. So it contains the potato extract. The boarded Django agar specifically contains the potato extract, and the Regan-Lowe medium contains the charcoal, blood, and the various antibiotics. So the pertussis is growth. Pertussis is cultured on the boarded Django agar or Regan-Lowe medium. Now let us discuss about the Telluride agar and Floss medium. The Tellurate Agars and the Floss medium are specifically used for Clostridium diphtheria. The Lowenstein-Jensen medium and the Middlebrook medium are rapid automated growth cultures. So these are the special cultures requirements that are needed for the Mycobacterium tuberculosis. Lowenstein-Jensen medium is the most used culture media for the Mycobacterium tuberculosis. The Lowenstein-Jensen medium, LJ medium, we call it Lowenstein-Jensen medium. We also call it as a LJ medium. The LJ medium contains the mineral salt. You have to take in mind that the LJ medium contains the mineral salt, asparagine, glycerol, malachite green, and hen's egg. So these are the various contents of the LJ medium. Now let us talk about Eaton agar. The main requirement of the formation of the Eaton agar is cholesterol. So the Eaton agar is required for the culture of the colonies of the Mycoplasma pneumoniae. So you can see in this picture also is that the colonies of the Mycoplasma pneumoniae has the appearance of the fried egg appearance. This is important, you should know. Now let us talk about the McConkey agar. The McConkey agar specifically allows the growth of gram-negative rot-shaped organism. That means the lactose fermenting enterics are cultured in the McConkey agar. The main content of this McConkey agar are the fermentation produces acid that cause colonies to turn pink. So as I have already said to you, the McConkey agar contains the pH indicator or lactose fermenter like E. coli will convert the lactose to acidic metabolites, which changes the color to pink. Talking towards the eosine methylene blue agar, EMB agar, we also call it the EMB agar. It specifically grows the E. coli, and the colonies with the green metallic sheen are seen that confirms the growth of the E. coli in the eosine methylene blue agar. Now there is one special culture media, that is charcoal is extracted buffered with cysteine and iron. So this is specifically used for the Brucella, Francisella, Legionella, and Pasteurella. So these are the 4 bacteria that is used for the growth. The charcoal yeast extracted buffered with cysteine and iron specifically allows the growth of Brucella, Francisella, Legionella, and Pasteurella. And now we have the special growth media I haven't mentioned here, but it is used to grow the fungi. We call it Severodestroc agar. The Severode agar, SDA agar, has the glucose and mycological peptones, and antimicrobial can also be added in the Severode's agar for the inhibition of the various bacteria. So SDA agar, Severode agar, this SDA agar will specifically allows the growth of the fungi. Now let us talk about the various type of the aerobes. So the aerobes are those type of bacteria that uses oxygen is their physiological need, and they use the oxygen to generate the ATP. The various examples of the aerobes includes the Nocardia species, Pseudomonas aeruginosa, Mycobacterium tuberculosis, and Bordetella pertussis. You have to know that the Mycobacterium tuberculosis is one of the important aerobic bacteria that we learn in the lung pathology, which causes the tuberculosis. So it usually resides the apices of the lung. The Mycobacterium tuberculosis bacteria reside the apices of the lung, because the apices of the lung is more ventilated than the lower part of the lung. So the reactivation of the microbial tuberculosis, especially after immunocompromised state or TNF alpha inhibitor use, has predilection for the apices of the lung, because the Mycobacterium tuberculosis is the aerobic bacteria and it usually wants to reside in the apices of the lung because of the more ventilation, more aerobic area. Now let us discuss about the anaerobes. The anaerobes are the bacteria which do not use the oxygen, is not dependent on the oxygen system to generate the ATP. So various examples of the anaerobes include the Clostridium, Bacteroides, Fusobacterium, Actinomyces israeli. So they lack catalase and or superoxide dismutase and thus susceptible to the oxidative damage. So these anaerobic bacteria usually generate the foul smelling wounds or foul smelling reaction due to the short chain fatty acids, and they are very difficult to culture and produce gas in tissue. The gas means specifically CO2 and hydrogen. These two are the gas they produce. So anaerobes are the normal flora in the GI tract, especially they are pathogenic in the elsewhere if they found outside the GI tract. So aminoglycosides are ineffective against the aerobe because this antibiotic requires oxygen to enter into the bacterial cell. So aminoglycosides are not used in the anaerobes. Talking towards the facultative anaerobes, the facultative anaerobes may use oxygen as the terminal electron acceptor to generate ATP, but can also use fermentation and other oxygen independent pathways for the generation of the ATP. So various facultative anaerobes are Staphylococci, Streptococci, and enteric gram-negative bacteria are some of the examples of the facultative anaerobes. Now let us talk about the intracellular bacteria. The intracellular bacteria can be divided into two, obligate intracellular and facultative intracellular. As the name suggests, the obligate intracellular are those type of bacteria that must reside inside the cell, but the facultative intracellular means that can operate outside the host cells also. So various examples of the obligate intracellular bacteria are Rickettsia, Chlamydia, Coxiella. They rely on the host ATP. Whereas the facultative intracellular bacteria are Salmonella, Neisseria, Brucella, Mycobacterium, Listeria, Francisella, Legionella, Yersinia pestis are the examples of the facultative intracellular bacteria. So these are the various explanation on the growth media. You have to know that growth media are specifically two types: selective media and differential media. And we discuss about the various special culture requirements, and we also discuss about the various bacteria that can be growth in this culture, and we discuss about the aerobes, anaerobes, and facultative anaerobes bacteria, and we also discuss about the various intracellular bacteria that can be divided into obligate intracellular and facultative intracellular. So this concludes our topic on growth media, special culture requirements, the various bacteria like aerobes, anaerobes, facultative anaerobes, and intracellular bacteria. I hope you understood this topic. Thank you.